Sorry. Okay. So welcome everyone. Thank you the organizer for accepting my talk. The a little view of my talk is a brief introduction of the TDA, how are related the phylogenetic tree with the horizontal gene transfer and the TDA, or talk about the my result about the with simulation and application with antibiotic resistant profiles. So the antibiotic resistance is one of the biggest problems now to the 2019. The global data attributable to the antimicrobial resistance is around to 1.2 millions by year. So antibiotic resistance is projected to cause to 10 millions to death annually to 2050. And the hospital are hotspot for multi-drug resistance and development, and horizontal gene transfer is a key to mechanism for antimicrobial resistance spread. For this reason, we interested to study new strategy to study the horizontal gene transfer. So, what is the TDA, or the topological data analysis? Is a technique of algebraic topology that consists in study the structure of the data sets. And there are many applications in many areas, how that graph, complex network, neuroscience, physics, metagenomics, and now genomics. In this figure, we can see the mapper algorithm. We can transform a data set to the graph, simple graph. So the object we study in TDA are the simplices and simplicial complex. We can see geometrically the simplices are the vertex, triangles, and edge, and tetrahedron, and so on. So the simplicial complex is the collection of the simplices that they can satisfy these two sentences. To if you have two simplices, the intersection is empty or is a face, or see you have the face of the simplex is other simplex. So in this figure is a geometric representation of the simplicial complex of dimension 2, and other thing, we need the elements stick together correctly. In the first figure, we have a simplicial complex. In the second, is not simplicial complex because the intersection there in object is not an element in the simplicial complex. So, okay, the persistent homology con the invariant topologies. For example, BT0 or BT0 con the number of the connected component, and BT1 con the number of the one-dimensional holes, or loops. We can think like loops. And for example, in this simplicial complex, we have three connected components, and we have two one hole. Sorry, here and here, because BT0 is 3 and BT1 is 2. So another definition, the filtered simplicial complex is a collection of the simplicial complex that we can think how evolve the simplicial complex. For example, we start with K sub 0, only 3 vertex, A, B, C, and in K sub 4, we have 3 vertex, 3 edge, and 1 triangle. And this collection of the simplicial complex is a filtered simplicial complex. So, for the previously simplicial complex filtered, we can calculate the Betty numbers, and the Betty numbers are For k sub 0 is 3, k sub 1 is 2, k sub 2 is 1, k sub 3 is 1, k sub 4 is 1. But in the BT1, we have 1, 1 in k sub 3 because in this time, we appear one holes. So how we can represent this information? We use the barcode, and the persistent barcode is a way to represent how a simple cell complex evolves. And the bars, the green bars represent the connected component, and the blue bar represent one hole. The length on the blue bar it represents the persistent, and a long bar indicate more significant features of my simple cell complex. So how we can relate this on phylogenetic tree and horizontal gene transfer? I try to explain this. So a phylogenetic tree, a record of evolution. The branch represent different species, and the nodes represent common ancestors. So when we use the persistent homology of the TDA, the tree or the theoretical data, so they don't have holes. Yeah, you can see the persistent barcode, this form. But when we have horizontal gene transfer and you apply the horizontal to persistent homology, you have one hole. So on. So we made many simulations. We start simulating a population in vertical inherence. We start with only ancestor. We fix the only 25 position initial set to 1, and in each generation we replicate this population, and we fix only one mutation for generation. When we apply the persistent homology at this population, we obtain that one persistent barcode with this, only one BT0. We don't have one hole. The second, we simulate horizontal gene transfer between three elements in our population, and we fix the length of the size on the groups. And when we apply the persistent homology, appear the horizontal gene transfer. In this case, we have 2 1 holes. So on. Now, when we add more groups with horizontal gene transfer, each group produce 2 1 holes. So now, when we change the length of the horizontal gene transfer, the persistence of the one hole changes. Okay? We have this figure when we move the length 0% to 20% of the length of the sequences with 2 transfers. So now we simulate 2 populations, vertical hierarchical. In the slide we have 2 populations, the pink and green. And the second figure, we simulate a horizontal gene transfer between elements the population 1 and 2. And when apply horizontal gene transfer, we have 1 hole here. And this is a geometrical representation of the simple cell complex, where we can see our 1 hole, and we have 2 sequences related in my horizontal gene transfer. So now I can skip this slide because Gabi all talking about this. So the clinical case, we study many bacteria isolate from USA hospital, and their three isolates, they have horizontal jet transfer. They have the same plasmid in F. And when we apply this strategy, the persistent homology, we detect two one-holes here and here, and in other words, we can see the geometric representation, my simplicial complex, and we can see the two one hole here and here. When the vertex appear, the isolate that have the horizontal jet. So, okay. The future direction applied to a longer data set of clinic isolate, applied to bacteria from Osas Rojas Patrocienega, probably, and DBLA automatize this strategy and other things. So one reference, and finally, thank you for attention and invitation for the BiGEvo 2025.
Thank you very much, Shaday. We have time for questions.
Shaday, I was wondering, which is the input, I'm sorry, for?
We can use the resistance, matrix of resistance, or we can use the genome and transfer this in a matrix, the present and absence matrix, probably.
That's very nice. In your simulations, when you simulate when you calculate the persistence, are you assuming that these genes are neutral, or would the persistence change if they were influenced, if they would help the microorganism, or on the contrary, it would be if they would be bad for it, and then some natural selection would be effective?
So, okay, only simulate change.
Yes.
Yeah, and we put one, we suppose this has genes, and zero she don't have a gene, only one in my simulation.
So persistence is the tend to disappear just with the same process, right?
Yeah, more the persistence is related with the how many times appear the one holes or the connected component. You have topological invariant.
Yeah, I think I kind of get it, but thank you.
Sorry.
We can talk later, maybe.
Okay.
Another question? Okay, I have one question.
Sure.
Can you use this tool, persistent homology, to study recombination in genomes?
Not yet, but I try to apply.
Could be possible.
Yeah.
Okay.